Genetics is the blueprint of life, and at its core lies the intricate dance of molecules that drive gene expression. This process, in which the information stored in DNA is used to produce proteins, is fundamental to how traits are developed, functions are carried out, and life itself is sustained. Today, we'll dive into the significance of gene expression and explore the remarkable process of protein synthesis, guided by the roles of DNA and RNA. Let's begin with the big picture: gene expression. Gene expression is the process by which the instructions encoded in a gene are used to produce a functional product, typically a protein. These proteins are the workhorses of the cell, responsible for everything from building structures like muscles to catalyzing chemical reactions as enzymes. Why is gene expression significant? It's the mechanism that bridges genotype and phenotype. The genotype refers to the genetic makeup of an organism, the specific sequence of DNA inherited from its parents. The phenotype, on the other hand, is the observable traits of an organism, such as eye color, height, or blood type. Through gene expression, the information in DNA determines these traits, shaping an organism's form and function. Now let's break down how gene expression occurs, focusing on the process of protein synthesis. Protein synthesis is a two-step process involving DNA and RNA, and it occurs in two main stages: transcription and translation. The process begins with transcription, which takes place in the nucleus of a eukaryotic cell. DNA, or deoxyribonucleic acid, serves as the template. DNA is a long molecule composed of smaller units called nucleotides. Each nucleotide consists of three components: a sugar molecule, deoxyribose, a phosphate group, and a nitrogenous base. There are four types of nitrogenous bases in DNA: adenine, A, thymine, T, cytosine, C, and guanine, G. These bases pair in a specific way: adenine with thymine and cytosine with guanine. During transcription, an enzyme called RNA polymerase binds to a specific region of the DNA called the promoter. It unwinds the DNA strands and uses one of them as a template to synthesize a molecule of RNA, specifically messenger RNA, mRNA. Unlike DNA, RNA is single-stranded and it contains the sugar ribose instead of deoxyribose. Additionally, in RNA, the nitrogenous base uracil, U, replaces thymine. The result of transcription is a strand of mRNA that carries the genetic information from DNA out of the nucleus and into the cytoplasm, where the next stage of protein synthesis occurs. This brings us to translation, the process of decoding the information in mRNA to build a protein. Translation takes place at the ribosome, a cellular structure composed of ribosomal RNA or rRNA and proteins. The ribosome reads the mRNA sequence in sets of 3 nucleotides called codons. Each codon corresponds to a specific amino acid, the building blocks of proteins. To help with this process, another type of RNA called transfer RNA or tRNA plays a crucial role. tRNA molecules carry amino acids to the ribosome, matching their anticodon, a sequence of 3 bases complementary to the mRNA codon, to ensure the correct amino acid is added to the growing protein chain. The process starts when the ribosome attaches to the mRNA and begins reading its codons. For instance, the codon AUG signals the start of translation and codes for the amino acid methionine. As the ribosome moves along the mRNA, tRNA molecules bring in the appropriate amino acids, which are linked together by peptide bonds to form a polypeptide chain. When the ribosome reaches a stop codon, UAA, UAG, or UGA, translation ends. The newly synthesized polypeptide chain then folds into a specific 3-dimensional shape, becoming a functional protein. Protein synthesis is a marvel of molecular precision. It ensures that the instructions encoded in DNA are faithfully translated into the proteins that carry out essential cellular functions. Without this process, life as we know it wouldn't exist. But gene expression is more than just a mechanical process. It's highly regulated. Not all genes are active at all times. Cells can turn genes on or off depending on their needs and environmental conditions. For instance, genes involved in digestion are only active when food is present. This regulation ensures efficiency and allows organisms to adapt to changing environments. Gene expression also explains how different cells in a multicellular organism can perform unique functions despite having the same DNA. For example, a nerve cell and a muscle cell have identical genetic material, but they express different sets of genes, resulting in specialized structures and functions. The implications of understanding gene expression are profound. It has revolutionized fields like medicine and biotechnology. For instance, researchers can develop targeted therapies for genetic disorders by correcting errors in gene expression. In agriculture, scientists use knowledge of gene expression to engineer crops with desirable traits, such as increased resistance to pests or improved nutritional value. In conclusion, gene expression is the vital process that brings the information encoded in DNA to life, allowing organisms to develop, function, and respond to their environments. Through the intricate steps of transcription and translation, DNA and RNA work together to produce the proteins that are the foundation of life's complexity. By studying gene expression, we gain insight into the mechanisms that shape living organisms and unlock possibilities for innovation and discovery in science and medicine.